Everyone here who's supposed to be. Four, five, six, seven, 8. Be 11. Thanks, everybody. Well, this is October 29th. We only have a few weeks left in this semester. Today we have an exam, lecture exam number 3, covering chapters 9 through 11. I admit I screwed up, and I copied this schedule from another instructor, and I changed some dates and stuff, but I didn't notice that we didn't have an exam covering chapters 7 and 8. So I think to fix that, I think next week, between now and next week, I'm going to put together a take-home exam on those 2 chapters. So it'll be an open book take-home exam, and should be take-home exam, open book. You should do well on it. So this would be something that could bring your average up. And also we'll cover those 2 chapters that we didn't on exams. I think in here we've been on the first 2 exams. We did the exam after break, right? And then you turned it in and you could leave when you were done. Is that what you guys like doing it that way? So I'll spend the first hour or so doing a little bit of lecture, and then we'll take a break, do the exam, and leave when you're done. So we covered chapter 12 last week, which was called Diversity of Life, but it had a lot of information about these evolutionary trees and the history of life on Earth, pretty much. And then taxonomy, classifying organisms into categories and giving them names, and explaining how to read an evolutionary or phylogenetic tree like this, and how they are created by scientists. Homologous and analogous structures. So next is chapter 13, and chapter 13 is in 2 parts in the PowerPoints, and I did not make these PowerPoints, and I'm not sure why the other instructor who did this did it in 2 parts, but they're both on Moodle. One was on last week's Moodle, and part 1 is on last week's Moodle, and part 2 is on this week's Moodle. But we'll cover both of them. Neither of them is very long. Change this. We're recording the right thing. Start the recording, right? Okay, so chapter 13, part 1, diversity of microbes. Microbes, or also called microorganisms, are organisms too small to be seen without a microscope, so you can't see them with the naked eye. Anything probably less than a half a millimeter long, we would call a microorganism or a microbe. So there are microorganisms in all three of the domains. Of course, bacteria and archaea, those are all they're all one-celled organisms. So all bacteria, all archaea are microbes or microorganisms. And even in the domain eukarya, which includes the animals, plants, fungi, and protists, it includes some microbes. Some fungi, there are some one-celled fungi. That's what yeasts are. Yeast is a one-celled fungi, and so they can be considered to be microbes or microorganisms. Some protists are one-celled, and they would be microorganisms. And there are even some animals that are, there are no one-celled animals. Part of the definition of animal is that they're made up of, they're multicellular. So there are no one-celled animals, but there are many of them that are so tiny that you would consider them microbes. And I'll talk a little bit about one that's especially interesting a little bit. As we've learned already, bacteria and archaea are prokaryotes. That means that their cells are prokaryotic. They have no nucleus or other membrane-bound organelles. And like we've also said, those two groups, bacteria and archaea, used to be considered together in one taxonomic group until just in the last few decades, comparing the DNA from those two groups showed that they're not closely related at all, and so they should not be in the same group. So they've been put into these different domains that we call bacteria and archaea. Some pictures here of some single-celled bacteria. It's hard to tell what some of these are, but this is one of the spiral-shaped bacteria that we'll talk about in a minute. Some bacteria are round like this. Some kind of fungi. This is a protist, red blood cells. So this is a parasitic protozoan. Talk about what that is. Hey, prokaryotes. Diversity just means a variety of different kinds of them. These were the first organisms on Earth. We think were prokaryotic. Earth formed about 4.5 billion years ago. Life didn't show up until a billion or so years later. Early conditions on Earth were nothing like the nice atmosphere we have now. There was very little free oxygen in the atmosphere, so we could not have existed back then. There was no oxygen to breathe. High levels of solar radiation, partly because the atmosphere just didn't filter out the solar radiation like it does now. There was a lot more volcanic activity, and that put a lot of harsh chemicals into the atmosphere. There was a lot of acids and hydrogen sulfide in the atmosphere all the time. So any organisms that lived in the first couple of billion years of existence on Earth had to be much different from now. They had to be adapted to those conditions. They for one thing, they had to be anaerobic, which means they did not require oxygen. There was none. There was no free oxygen. Probably where life originated on Earth was deep in the ocean, at hydrothermal vents where there's hot gases and chemicals come out of the Earth's crust, and the first one-celled organisms were adapted to use the energy at those hydrothermal vents as energy sources. The first, then phototrophic organisms. These were some early form of photosynthesis. Appeared, and these would have had, these were photosynthetic organisms, but they were anaerobic. So they could do photosynthesis, but there was not oxygen for them to use, but they would produce oxygen by photosynthesis, and so that's how the atmosphere changed, so these photosynthetic organisms took a lot of carbon dioxide out of the atmosphere and released oxygen. And for these organisms first lived in water, they lived in oceans. So they added oxygen to the ocean water until the ocean water became saturated with oxygen, and then the oxygen diffused into the air, and eventually it changed the atmosphere so there was enough oxygen for aerobic organisms that required oxygen to live. So these early photosynthetic one-celled organisms were similar to the cyanobacteria that we have now, which are photosynthetic bacteria. Here's cyanobacteria living in Yellowstone National Park, and it says they're green. The word cyanobacteria comes from this color. They're a color called cyan, right? I think it's a bluish green color. And in fact, these cyanobacteria, they used to be called blue-green algae. It was a common name for them. But that was a bad name because they're not algae at all. They're bacteria. Algae are protists. They're not bacteria. So there are these cyanobacteria living in this little stream in Yellowstone National Park. Because of the hot springs, and also a lot of chemicals, sulfur chemicals that are in these hot springs and geysers in Yellowstone Park. It's an unusual environment that has a lot of organisms that don't live in many other places. You wouldn't see cyanobacteria like this in a stream flowing through Richmond County, probably. They are in Yellowstone. These microbial mats are just layers of there were dense populations of these early microbes, and they were probably originated around hydrothermal vents in the ocean, they use chemicals in these hydrothermal vents for food. They used a process that's something like photosynthesis, except it's called chemosynthesis. Instead of using sunlight for energy, they would use chemical energy in some of these molecules in these hydrothermal vents, like hydrogen sulfide. They would use them for producing their own food, something like photosynthesis, but without light. These stromatolites, stromatolites are just layers of it's like sand particles glued together that are believed to be formed by a lot of these early one-celled organisms that secreted secreted a slimy glue type chemicals that glued all these sand particles together, and so now there are these hard crusts that have been found, and so they are believed to be fossil evidence of these early microbes that lived in these colonies in some places. Extremophiles are microbes that live in extreme environments, and there were these early one-celled organisms that lived in hydrothermal vents. That's an extreme environment. Water coming out of the Earth's crust with these harsh chemicals in it. But there are organisms that they thrive in environments like that, and they still exist. There's plenty of extremophiles, mostly archaeans, that live in very hot places, like hydrothermal vents or mineral springs in Yellowstone Park, and some of them like very acidic environments, and some like very salty places like the Great Salt Lake in Utah, which is saltier than the ocean. There are archaean one-celled organisms that live there, and they can't live anywhere where it's not that salty. They love it. These pictures don't look like much, but this one on the left is a microbial mat of like a accumulation or colony of one-celled organisms at a hydrothermal vent in the bottom of the Pacific Ocean. And then on the right, stromatolites, so these rocky crusts that are left over from where there used to be these thick layers of one-celled organisms. This term biofilm just means it's just like a slimy layer that has a lot of microorganisms in it, and these are everywhere. There's it can usually in wet places. Just a rock in a stream would have a biofilm of it, of microorganisms on it. Inside pipes and sewage treatment plants, surfaces in your house, and even inside your mouth. You wake up in the morning, you got a biofilm in your mouth. You've got a community of bacteria that living in there. That's why your mouth tastes weird, tastes bad in the morning, because of those bacteria that have grown there overnight. Any place where it's wet and there's something they can eat, bacteria will grow. They'll grow on your surface of your eyeballs, except that there are antibacterial chemicals in your tears that prevent that. Yeah, so that's the reason to brush your teeth in the morning, even if you didn't eat breakfast, because you've got all that bacteria will do damage. Prokaryotic cells, they don't have a nucleus, and they don't have all these other organelles like mitochondria and chloroplasts and endoplasmic reticulum like eukaryotic cells have, but they have pretty much the same processes going on inside those eukaryotic cells. They just don't occur in these little contained within organelles like in human cells or other eukaryotic cells. As we've already learned, all cells, whether they're prokaryotic or eukaryotic, have a plasma membrane surrounding the whole cell. They have cytoplasm, which is just the stuff inside the cell. And they have genetic material, chromosomes of DNA. If it's a eukaryotic cell, like human cell, there are some number of long straight chromosomes. In humans, it's 23 pairs. Other species have different numbers. While in prokaryotic cells, like bacteria, they have just one chromosome usually, and it's a loop of DNA. And they also, all cells also use RNA and they make proteins using the information in the DNA. And then all cells have ribosomes because all cells have proteins. I keep saying all cells. There are some exceptions to almost everything. There's one kind of human cell that doesn't have all these things. Your red blood cells don't have chromosomes. Red blood cells don't have chromosomes. They don't have ribosomes. Early on, when red blood cells are first formed, they have chromosomes and ribosomes for making the hemoglobin protein that carries the oxygen. But once those cells are formed, the chromosomes disappear, the ribosomes disappear, and then for the rest of the red blood cell's life, all it does is carry oxygen. It doesn't do a lot of the processes that other cells do. They don't have chromosomes, so they can't even reproduce. They have no information for doing anything. So all they do is it's like a membrane surrounding a bunch of hemoglobin for carrying oxygen. They live for a few minutes and then they die. So not all cells have all this stuff. Prokaryotic cells come in three major shapes. Bacteria, and I'm not sure about archaea. I think so, but bacteria come in three shapes, and they have these names: Coccus, Bacillus, and Spirillum. Those are the plural. Coccus is plural. Bacilli, Spirilla is plural. But so they're round, rod-shaped, or spiral. So the Coccus bacteria is round, and the Bacillus kind are long, like rods, or look like little hot dogs or something. And the Spirillum are spiral, like little springs. Some bacteria, the names of some bacteria have those words in the names, like may have heard of Streptococcus or Staphylococcus. Staph and strep, those are infectious bacteria. And those bacteria are round. It tells us in the name, Streptococcus, which means round, or sphere. There are some bacteria have the word bacillum, bacillus in there. one called Bacillus thuringiensis, bacterium that kills insects and it's used as a pesticide. So here's what they look like under a microscope, and these are very tiny. Bacteria cells are way smaller than most human cells, so you need a really high magnification, high power microscope to see them as well. So this, of course, would be the coccus and bacillus and spirillum, microscope spring. There's one bacterium, one involved with sexually transmitted disease, syphilis, spirilla bacterium. The way I remember, spirilla, that's one easy. That one's easy. Spiral shape. Remember that one. Coccus and bacillus, which is round and which is thoracic. I remember coccus has an O in the word. It's the round one. Bacillus has two L's in it. The long, rod-shaped letters. So if you take a course in microbiology, you get a lot of time learning about bacteria, different shapes of bacteria. The characteristics of bacteria, and we've already learned this in previous chapters. They have a cell wall. The cell wall in plants is made of cellulose, of polysaccharides, but cell wall in bacteria is mostly this stuff called peptidoglycan. It's a combination of carbohydrate and protein together. Archaea cells also have cell walls, but they're not necessarily made of this peptidoglycan. And this, some cells have more peptidoglycan than others in their cell walls. Some antibiotics. An antibiotic is a drug for killing bacteria, killing infectious bacteria, or disease-causing bacteria. And many antibiotics kill bacteria by they prevent the bacteria cell from making this stuff. The bacteria cell can't make a cell wall, it'll die. So some bacteria have more of this stuff than others, and that type of bacteria cell will be affected by a particular antibiotic. Some bacteria have a capsule around it. It's just an added layer of material in addition to this plasma membrane and the cell wall. They have this third layer of stuff. In the nucleus, they don't have a nucleus. There's no membrane surrounding the chromosomes. So wherever the chromosome is located, it's just called a nucleoid. And some bacteria have a flagellum, or some might have two, usually not more than two flagella on bacteria cells. Most of them don't have a flagellum, but some of them do. And bacteria, we tend to think of bacteria as being bad. They're germs that make us sick and cause diseases, but the vast majority of bacteria are perfectly harmless. Can't hurt you at all. Really, relatively few of them can cause diseases in humans or any other organ. They reproduce by splitting in half. So that binary fission. Binary means two, fission means to split in half. So this is, we don't call this mitosis, mitotic cell division, because in mitosis there's prophase, metaphase, anaphase, and telophase. The chromosomes line up and then separate. But in these, there's only one chromosome. So there can't be a prophase, metaphase, anaphase, and telophase. There's no nuclear envelope to disappear and prophase or anything. So let's call it binary fission. But it's like in eukaryotic cells before the cell divides, it has to copy its one chromosome so that there will be a chromosome in either daughter cell. Then the cell gets longer and then it just cell wall grows between the two daughter cells and like the two daughter cells would be identical to the original cell unless there was a mistake, a genetic mutation. For example, when the chromosome is replicating, being copied, if there's a mistake, then the chromosome in one or both daughter cells would be different. That's how genetic variation occurs in bacteria. Some bacteria can reproduce really fast, so a generation could be 20 minutes. The bacteria, one cell divides, now the daughter cells will be ready to divide 20 minutes later. So that's how populations of bacteria can explode really fast. After only, if you start with one cell, after only 10 generations, you're up to 1 million cells already. After 20 divisions, there's over 1 billion cells. Happens every 20 minutes. That doesn't take long at all to be up to 1 billion cells. There's no genetic recombination because there's no sperm and egg coming together. It just divides in half. But here's a genetic recombination does occur in prokaryotes in these three ways, but this PowerPoint doesn't explain what those mean. But the transformation transformation is something that is refers to something that bacteria can do. It just means if a bacterium finds some DNA in the environment, if there's bacteria on this tabletop, going along and there's some DNA there, just maybe because somebody was leaning on it and left some skin cell. Bacteria find that DNA, they can pick that DNA up inside it, and put it into its own chromosome. DNA and say, hey, cool DNA, maybe I can use that. Pick it up inside, and now it has a bunch of new genes that maybe it can use those genes to make some kind of protein that might be useful to it. It's called transformation. Now, transduction is another way that, it's a way that DNA can get from one bacteria cell into another. If there are viruses called bacteriophages, bacteriophage is a virus that only infects bacteria. It can't hurt you at all. It just infects bacteria. And when these viruses gets into a bacteria cell, then that virus will take some bacteria DNA into the virus DNA. Now that virus infects a another bacteria cell, it can now transmit that bacteria, that DNA from the first bacteria cell into the second one. It's called transduction. And a third way that bacteria cells can gain or give DNA is called conjugation. So here's a couple of bacteria cells. One of them can here's its chromosome, DNA, and it can make a copy of some of its DNA. It might make a copy of this section of DNA, and then it'll send a tube out to another bacteria and send that piece of DNA in there. So here, you want some of my DNA? Maybe you can use this. And that's called conjugation. This kind of thing, way that bacteria cells can share genes. I mean, other organisms can't. We can pass on our DNA to our kids, but I can't give my DNA to you. I can give a gene to you if I want to. People ask me, you have good genes. I have some. But and this is causes problems. If there's a kind of disease-causing bacterium, and scientists come up with an antibiotic that'll kill that kind of bacteria, and it's working fine, curing diseases for years. But there are other bacteria that will resist that. They have genes that make them resistant to that kind of antibiotic. So now the disease-causing bacteria can get that gene through one of these processes. Get that gene to make them resistant to the antibiotic. Now that antibiotic doesn't work anymore. So in medicine, has to always be coming up with new antibiotics because the old ones stop working. Partly because of this kind of thing. Most microbes are harmless to humans, but some of them are pathogens. A pathogen just means an agent that causes disease. The word pandemic, of course, we know that word lately. Pandemic is just a worldwide epidemic. An epidemic just means an epidemic is a spread of a disease through a population. If it's the worldwide population, we call it a pandemic. There have been many epidemics and a few pandemics. Through the bubonic plague, killed a large percentage of the European population, spread by fleas that lived on grass. Food poisoning, there have been some outbreaks of food poisoning caused by certain bacteria like E. coli, Escherichia coli, abbreviated E. coli, is one that occurs normally in the colon of animals, including humans. But some strains of it are produce toxins. If it gets into food, like this, they don't use the right sanitary practices, water houses where they dump animals. Now the E. coli in the meat, the meat isn't cooked properly, kill the bacteria, and it can be fatal. People have died from it. There have been Jack in the Box restaurant once killed several people serving contaminated meat. there's a case of some kind of organic apple juice. Organic means um, they not grown with pesticides, and they, they did not pasteurize this kind of this strand of apple juice. Pasteurizing means simply they heat it up to kill any bacteria. But this was supposed to be all natural, organic apple juice, and so they didn't treat it anyway to kill bacteria, and it ended up killing a bunch of little kids. And it turned out that these apples, these orchard workers who were to pick the apples off the trees, they were picking some up off the ground, and there were deer that were pooping on the ground, so the bacteria from the fruit was getting on the apples. And the apple juice was contaminated with bacteria. So people thought they were buying this healthy, natural apple cider, when in fact it was deadly bacteria. Antibiotics, they're drugs to kill infectious bacteria. Before antibiotics came along, it was in about 1940. Before that, if you got an infection, you could die from it. Die from it. Just any simple infection. Antibiotics just completely revolutionized medicine. Because they're so effective at killing infectious bacteria. And the first one was penicillin, and a scientist named Fleming discovered he was deliberately growing bacteria in petri dishes. He was a microbiologist, I guess. And he noticed that there'd be a little colony of kind of mold growing in a petri dish where he was trying to grow bacteria, and there were no bacteria around that colony of penicillin. So it turned out to be molds were producing penicillin. So he was able to figure out what it is that this mold was producing. It was preventing the bacteria from growing. He isolated that chemical, and that's what became the first antibiotic, penicillin, which is still used a lot. So, very important antibiotic. But many bacteria that are resistant, that early on were effectively treated with antibiotics, they have become resistant to antibiotics. And this well-known one is this one called MRSA, methicillin-resistant staphylococcus. So this is the name of the bacteria, scientific name, Staphylococcus aureus. This is a common bacterium that's everywhere. For some reason you want to kill, you want to clean your bathroom because this stuff's growing everywhere. Well, I think at any given time, 30% of people have staph bacteria on their bodies. And if it gets into a spot or something, it can cause an infection. So the methicillin is a very effective antibiotic. It kills a lot of different kinds of infectious bacteria. But there are some staph bacteria that have become resistant to methicillin and most other antibiotics. And this is a cause of it's believed that this happens as a type of natural selection. So not just like in any other organism, not all staph aureus bacteria are genetically identical. Different genes. And some of them just happen to be maybe there's just a few of them out there that have some genetic resistance to antibiotics. And so people would get a staph infection, the doctor would give them antibiotics for it, and that antibiotic would kill off all the staph except a few staph bacteria cells that would survive. And then those would live on in the person, and now that person has a population of staph bacteria that are all resistant to the antibiotic. So that's a type of natural selection. Ones who didn't survive the antibiotic, they died, but the ones who survived had a gene that made them antibiotic resistant, and they developed babies. Their offspring now have that gene for resistance to the antibiotics. So this is one reason that we're told if you get prescribed antibiotics and it says take it for 2 weeks, don't stop if you feel better after 5 days. Take it for the whole 2 weeks, because if you stop after 5 days, then there might be some that are resistant enough, but they're still alive. 99% of them are dead. 1% that was tough enough to survive 5 days of antibiotics is still alive. But if you take the whole 2 weeks, that's the one that might kill them off. And this is also why you don't really need an antibiotic. Don't take it. Doctors, in the past, doctors used to prescribe antibiotics too much. You go to a doctor that's old and he prescribes an antibiotic, even though he knows that antibiotics kill cold virus. Antibiotics kill bacteria, not viruses. But the doctors used to do that just because they know that patients who come to you and they're sick, they expect you to give them some medicine. They just prescribe an antibiotic, even though they know it's not going to help. So that indiscriminate use of antibiotics too much just encourages this natural selection that causes antibiotic resistance. So this is a big problem. All these antibiotic-resistant bacteria, and medicine, pharmaceutical companies have to keep coming up with new antibiotics to keep up. Okay, so already talked about how some bacteria use certain kinds of fermentation that produces alcohol or lactic acid that for making different kinds of food and beverages. Bacteria are used in biotechnology to produce some kinds of molecules, antibiotics, and then this term bioremediation, remediation means finding a remedy for something. So bioremediation means using living organisms to solve some kind of problem that we have. And one important type of bioremediation is using bacteria that actually eat petroleum oil. And this is important in oil spills. There was a big oil spill in Alaska, Exxon Valdez, that was, I don't know, 40 years ago or so. It was the biggest oil spill in history up to that time. And a lot of birds and marine wildlife was killed, and this whole big area, most of Alaska, was covered with oil. But it was big news for a long time. They were upset about it. And there was people were trying a lot of different ways to try to clean up all this oil, and it turned out that that the natural bacteria in the environment over time eat the oil and break it down, and it took a long time. But, if we can come up with really efficient bacteria that eat a lot of oil, I mean, we could deliberately put those bacteria onto oil spills, and we could speed up that natural bioremediation. A lot of bacteria and archaea in and on our bodies. In fact, you have more non-human cells in and on your body than human cells. There's bacteria that live. And how much you bathe, you might think you're really clean, but you're really covered with bacteria. And mucous membranes in your airways, and part of your digestive system, especially the large intestine. You have a huge population of bacteria in there. They're supposed to be there. If you don't have this normal population of bacteria, or much of the wrong kind live in there, and not enough of the right kind, or some imbalance, you can be sick. There are people who have chronic digestive problems, like chronic diarrhea. It just goes on for years. So recent research is finding out that that's often caused by they have the wrong bacteria in their large intestine. There's one kind of intestinal bacteria that's called, I forget the full name, but it's abbreviated C. diff. We call it C. diff. And this is one bacteria that's in most of us, but if you get too much of it, too much of this kind, it can cause chronic digestive problems. There's a treatment for this now that's still mostly experimental, but it seems to be working. It's called a fecal transplant. Putting food from one person into another helps to cure them of chronic problems that are caused by wrong bacteria. And they actually will take the feces from a healthy person and put them in the capsules, and then have the patient swallow them, and letting the bacteria from the healthy person populate the colon of the sick person. And there have been some cases of miraculous curing of problem. There was one woman who had this C. diff problem so bad she couldn't leave the house. But she had to poop every 20 minutes. She couldn't be able to get away from the bathroom. And she had a fecal transplant after years of these symptoms, and within a month she was perfectly normal. Okay, so there's that Valdez. This was a big oil tanker owned by the Exxon oil company that split open off the coast of Alaska. Eukaryotic organisms. Like I said, the eukaryotes were the first. Were here first. About 3.5 billion years ago. Then the eukaryotes didn't appear until almost 1.5 billion years later, about 2 billion years ago. And we already talked about the theory of endosymbiosis, how the first eukaryotic organism, how the organelles occurred. The first mitochondria and chloroplast were believed to be separate cells that got into other cells and then became organelles. Endosymbiosis. And evidence for this is chloroplast and mitochondria, which are these organelles that are believed to have started out as other cells that got into cells. They have their own DNA, which is separate and different from the DNA in the cell nucleus. And they have a double membrane, layers of membrane around them, and that's believed to be the cause that if a cell entered another cell, and that cell first had a plasma membrane around it, but as it pushed its way into the other cell, and the plasma membrane of that other cell wrapped around it and formed a second membrane. And that double membrane layer still exists in the chloroplast, mitochondria, and cells. And also chloroplast and mitochondria within cells. When if some of your cells need more mitochondria, the cell makes more mitochondria by the mitochondria just divide. It's like cells dividing. So these mitochondria and chloroplast, they still act like separate cells that live inside of bigger cells. Some pictures of mitochondria. Just diagram showing the endosymbiosis. Cells getting inside of other cells and becoming organelles. Okay. Wanted to real quick talk about a couple things before we leave this topic of microbes. Oh yeah. These things. These critters called tardigrades. Weird looking things. They have 8 legs, little claws on them. They're called water bears. Water bear is a common name. So they look like kind of like little bears, but they're tiny. The typical tardigrade is maybe less than a half a millimeter long. So you never see them unless you were looking for them, put them under a microscope. We can probably find some if we go around and find some moss. They like to live at moss, but they live in a lot of different places. There's a couple thousand species of these things. But what's interesting about these guys is that they're almost indestructible. They can survive. It tells us here they can withstand temperatures as cold as minus 328 degrees Fahrenheit or as high as 300 degrees. Way hotter than boiling water. They can survive that. They can survive pressures of 6 times the pressure in the deepest part of the ocean. They can survive the vacuum of outer space. They can be completely dehydrated and dried out and come back to life. It's amazing. They can survive the gamma rays, which are real high energy electromagnetic radiation. So you can't kill these things. So saying that if something happens, wipes out humans on the planet, these guys probably still be around. One other thing I want to talk about real quick, because we're in the middle of this. I don't know if we're in the middle or at the beginning or closer to the end or the beginning of this pandemic. It seems to be getting worse. Our president tells us it's over. Last spring, I put this extra credit assignment. I was taking this extra credit assignment with some of my biology classes. I just wrote out this paragraph about describing the COVID-19 virus, but it's actually full of lies. Almost everything in this is wrong. But that's the assignment is telling you what's wrong. So the first sentence: all viruses are harmful to humans. That's not true. Most viruses are completely harmless to humans. So that first statement is a lie. The COVID-19 virus consists of disease-causing cells. The virus is not a cell. Virus is just DNA or RNA surrounded by a protein capsule. That's all there is to it. Not a cell. They get into the human body and eat away at the tissues of the rest of the solar system. They don't do that at all. Viruses don't eat anything. That's not true. These tiny living organisms, viruses are not living organisms. So they're not cells. The cell theory tells us every living organism is made up of one or more cells. They're not cells, they're not alive. There's no metabolism going on in the virus. They can't even reproduce themselves. They infect a cell, and then the cell makes copies of the virus. So they're not living organisms. They can reproduce outside of the body. They can't reproduce on their own at all. So the more they eat, they don't eat. The more they eat, the faster they reproduce by cell division. They're not cells, so they don't divide by cell division. Biomedical researchers are trying to make a vaccine that would be an injection of antibodies. The vaccine is not an injection of antibodies. The vaccine is something that causes your body to make antibodies, but it's not an injection of antibodies. Because antibodies, which are special white blood cells, they're not the protein molecules that are made by now, when we eat viruses, antibodies don't eat anything. Just protein molecules that grab onto to a virus to disable it. There's like nine lies in here. And I was going to try to assign. I actually had some students who, after they were at the end of their, they've taken 2 whole semesters of human anatomy and physiology, and they told me everything on here is true. It's all correct. Couldn't find anything wrong about that. That's disappointing. Okay, well, let's take a break, and we'll do this little exam.